Okay, good morning, everybody. Today we're going to talk about pharmacogenetics and pharmacogenomics. Okay, this is a very important field for all of us because it's been realized that genetic differences among us will cause us to respond differently to therapeutic drugs in such a way that either the drug may not work for you because you have a different metabolism from some people, or it will give you too high of a dose, in which case it might be toxic. So DNA testing can tell who is which, but lots of times these tests are not offered. So this whole collection of adverse drug responses is really something that we all need to be concerned about. Generally, thinking about therapeutic drugs, what they do is they, of course, bind to specific proteins and either inhibit or enhance them. And it's usually something that's important, crucial to the disease. But what's important to realize is that when the drug enters the body, many things have to happen to it before it can actually take therapeutic effect. Many times, drugs are administered in such a way that they need to be acted upon, either to activate them, but also there's also a process where the drug is inactivated and excreted. And it's the balance between those two processes, activation of a prodrug, which we'll get to in a sec, and clearance from the system, that determines the therapeutic level that a person will experience. And this varies enormously among people. Okay, this has become a major thing for many people, and there's actually a really important website which does have prescribing and dosing information for many of the drugs that are currently in use, because it's become widely realized that genetics plays a huge role in what's going to work for somebody or be toxic to them. Okay. Pharmacogenomics, it's a big thing now, and this implies that. Okay, one important and very relevant idea here is that of therapeutic index. You may not realize that many drugs are toxic in higher amounts, and that normal dosage, or what's considered the average dosage, is dependent on the metabolism by average people. And you may not be average, and you may be harmed by normal levels of harmless drugs, or else the drug may not work for you at all. But the therapeutic index idea is something, and here's an example. It's a therapeutic index for a particular blood pressure medication. And what's shown here, don't worry about the units, is the red curve describes the amount of this particular blood pressure drug that has a therapeutic effect as a function of its concentration. The green shows what will happen, or the level that it takes to actually be toxic for this particular drug. Now you want the therapeutic index, which is to say the effective dose that's going to help you, to be very different from the toxic dose, which is going to hurt you. And you can see that those two curves are reasonably far apart, and the therapeutic index here is roughly 100, so it takes a lot more of the drug to harm you than is effective. So this is something like a good drug. And what you don't want is for the toxic effect and the therapeutic effect to be close to each other in concentration. And add this to the fact that different people have different levels of toxic effect, this becomes an important consideration. Okay, so the idea is that drugs are metabolized by the body, and sometimes, often, it's necessary to activate the drug. So what's given as a compound, or what's given to the person, is not the active form of the drug, that the drug actually has to be acted upon metabolically in the cell before it can actually do you any good. So that's what's called a prodrug, where it's actually metabolized into its active form in the body. So in this case, if a person happens to lack the enzyme or be deficient in the enzyme that activates the drug, then this drug won't benefit them. It won't work, or it will work much less well than it would for somebody with an active enzyme. You also have the opposite effect, where you have to actually inactivate the drug for excretion from the body. So the body metabolizes it for removal. Now, some drugs are toxic if given in too high doses because of the therapeutic index. So that means if your rate of inactivation is too slow, then there's going to be a problem, and there can be a very fine line between what's effective for somebody and what's toxic. So the ultimate level of drug in the body over time is going to reflect a balance between those two processes, where the drug is activated to become active, or inactivated and excreted. And we'll have some examples in a minute. Okay, so we have to think about the drugs being cleared from the body and inactivated. And in summary, the amount of drug that's actually active depends on how quickly the drug is removed, but also how quickly it's converted to the active form. And there is a great deal of variation in humans as far as how we respond to the various drugs. And this, of course, has very important clinical significance because those two competing reactions will determine how much of the active or possibly toxic drug is present at a given time. Okay, so the types of reactions that can happen various, including methylation, acetylation, sulfation, etc. And we have enzymes that perform these reactions. These can be either activating or inactivating modifications. There's also a lot of various oxidation, reduction, hydrolytic reactions, etc. So there's a lot of metabolism that meets most drugs that are placed in the body. All right. So the idea summary is that people vary a lot in how they metabolically deal with the drugs. So what's a good safe dose for somebody else might be very toxic for you. Or else what's normally a helpful therapeutic dose might not help you at all. Importantly, these are genetically determined and can be tested for before you are exposed to a potentially lethal dose, or worse, given a drug, well, not worse, given a drug that will not work for you. Okay, the classic example of this has been around for a long time, is the example of a particular drug called 6-mercaptopurine, which is administered as a prodrug, as you'll see in a second. This is a very important drug for use in childhood leukemias, among some other cancers. It's usually the drug of choice, really, for things like childhood leukemias in general. Okay, what this drug does is inhibits DNA replication, has several methods for doing that, and the fact is that cancer cells are more sensitive to this because they're replicating very rapidly. And it was realized early on that some children are made very sick by the drug, whereas most people do fine. And this has to do with a genetically determined metabolic handling of this molecule. Okay, 6-mercaptopurine, anti-cancer drug, and it is an example of a thiopurine. So the sulfur here on the purine ring replaces the oxygen that would normally be found on a guanine. Now, in and of itself, this molecule is not active as an anti-cancer agent because it needs to be converted to the active forms, which include the thioguanine nucleotides, which include the sulfur instead of the oxygen. Okay, so here's the oxygen, the guanine has an oxygen, 6-mercaptopurine has a sulfur. Okay, so there's two things that can happen, and it's the balance between those two that determines the level that's present in the body and determines whether something is going to be toxic to somebody. Okay, so the first thing that happens is you have a balance, and the important enzyme here is the TPMT methyltransferase, thiopurine methyltransferase. That enzyme is responsible for disposing of the thiopurine. That's what's going to inactivate it and remove it from the body. So people do inactivate this at a reasonable rate, and that clears it from the body because it is toxic in higher amounts. Okay, the opposite effect, of course, is to activate it to form the active form, which is thioguanine nucleotides, and this is what disrupts cell division. So when the person is given the 6-mercaptopurine, both of these reactions will occur, and they're competing with each other. We're going to dispose of some of it immediately because of our TPMT enzyme, but we're also going to activate it so that it's able to then carry out its function to inhibit DNA replication and cell division. Okay, so in terms of activating it, the active form is really these thioguanine nucleotides, which are basically look like nucleotides, except that they have a sulfur where the O is supposed to be in guanine. So, that's the activating process, and the inactivating process is where we simply clear it from the body by methylating it and then disposing of it. Okay, now, the assumption is always that these processes compete with each other, and that the rates of those reactions are the same in everyone. Well, that is far from the truth, because some people are very deficient in this TPMT enzyme, the thiopurine methyltransferase. And in those people, what happens is you end up with too much of the thioguanine nucleotides because it's not disposed of properly. So extra amount accumulates, and that's why the kids become sick. Okay, so the clearing activity competes with the one that drives the thiopurine into metabolism, which is intended to lead to the death of the proliferating cells. But the balance is important. You need enough to go into metabolism to have a therapeutic effect, but we also are counting on the fact that some of it will be degraded instead. And if the rates of those two reactions are very different from each other, that's going to cause a problem for some people. Okay, we didn't go through, this is the mechanism by which the thioguanine nucleotides inhibit replication and repair. Read this if you want to. Okay, so genetic heterogeneity in humans leads to different responses. Okay, the important feature here is that the thiopurine methyltransferase activity differs a lot among individuals, and this causes problems if they don't dispose of it in an average way and they're not tested for it. Okay, so what happens, of course, is that people that are deficient in this enzyme will not inactivate the drug very quickly. It will not be cleared from their system at expected rates. And that's what's going to happen is they're going to build up toxic levels of thioguanine nucleotides. When this amount is too high, it is toxic and can be lethal in this case. And of course, the reason is if you're not clearing the compound from the body, the extra will form the thioguanine nucleotides, which are toxic if there's too much of them. Okay, so here's a diagram showing the activity variation in people, and what's being plotted is the amount of thiopurine methyltransferase activity on the x-axis and the number of amount of people that have that particular level. So you can see it's kind of a bimodal distribution. We've got a big peak around maybe 12 or so units in red blood cells, and there's a distribution around that average. But then we have a cluster of people between 5 and 10 who have substantially lower amounts of this activity in their blood cells, implying that there is a difference in this subpopulation of cells, of people, relative to how much of this particular enzyme that they have. And this will influence how they will respond to the 6-mercaptopurine if they get leukemia. So the enzyme levels are correlated with the thioguanine nucleotides. And in this particular graph, we have the enzyme level on the x-axis and the thioguanine nucleotide on the y-axis. And you can see that they're inversely correlated. People with low amounts of the TPMT enzyme do end up with higher amounts of the thioguanine nucleotides, sloping down to people with a lot, 20 units, have much lower levels of the thioguanine nucleotides. Now, this is what you'd expect if you have competing reactions. If you have higher enzyme level, you will be driving more of it out of the body. If you have lower enzyme level, you're going to be creating more thioguanine nucleotides than average. And most people are kind of scattering around in the middle of that range. Okay, so that makes a lot of sense, that we will have some people with lower amounts of the TPMT enzyme will, because they are not carrying out the degradation reaction, will have higher thioguanine nucleotides. So those processes are inversely related to each other. All right. So if you don't dispose of it rapidly, you're going to accumulate more, and the drug can be built up to toxic levels. Okay, so here's a pseudo-clicker question. Compared to normal, people with 5 to 10 units of TPMT will have elevated thionucleotides, reduced thionucleotides, similar amounts of thionucleotides. Well, the answer, of course, is A. If you're not disposing of it, then you're going to have more driven into the active form, and so you will have elevated thionucleotides. Okay, this makes sense. Same picture. A person who gets sick from the drug is most likely to have low TPMT enzyme and low thioguanine nucleotides, low TPMT enzyme and high thionucleotides, high TPMT enzyme and low thionucleotides, high TPMT enzyme and high thionucleotides. Okay, that was a tongue twister. The answer is B, of course. If you have low degradation, you're going to have higher accumulation of the active and eventually toxic compounds. All right, so the toxic compounds are thioguanine nucleotides. People with less of the TPMT enzyme will have more. This makes sense because the TPMT enzyme reduces the amount of therapeutic compound that is formed. So if you've got more of this enzyme, you will have less of the thioguanine nucleotides. Yes. Right. So this is where genetic testing comes in. And people are actually tested for this particular thing because it's been known for so long. And it's very well characterized in terms of what's happening. So this actual DNA test is generally offered to people that are considering being prescribed to take this particular drug. So this is a good case because it's well known and people are tested. So the toxic effects are generally avoided. Okay, so here's a picture of the polymorphisms, the most popular, most common alleles that are out there. And drug people use a strange kind of nomenclature, which we have to get used to. It's the TPMT asterisk 1 is the wild type form. It's considered the normal form, and that's the form of the enzyme that most people have. But then there's some variants out there. TPMT star 2, TPMT star 3A, 3B, 3C, 3D, and some others. So the most important ones that are figuring in this are actually the star 3 series of mutations. And these are well characterized. Now there's some other ones that are well known now, but their effects on the enzyme aren't quite as well known. Okay, so the nucleotide substitutions are known exactly at what position they occur, and they define these 3A, 3B, and 3C alleles. These alleles produce very little enzyme activity, and people that are homozygous for these inactive enzymes are extremely sensitive to thiopurine drugs because they don't dispose of them efficiently. Okay, there are amino acid changes in each case. Okay, and there are more alleles now, but these are the most frequent ones, and those are the ones that are known to cause a problem. Okay, so this can be complicated. What's fairly horrifying is actually, well, the people that are homozygous are relatively infrequent in the populations, approximately 1 in 300 Caucasians. But the scary part here is the middle part. People who have one normal gene and one gene variation with decreased, which is those heterozygous people in the previous graph, that they will produce an intermediate amount of activity. And this level is low enough that 30 to 60 people of those heterozygotes will have severe side effects. So even though the homozygotes are relatively rare, not very rare, approximately half of the people who are heterozygotes, who are not rare, will have side effects. People that have 2 copies of the variant homozygous, they will have a serious likelihood of developing severe bone marrow toxicity. And so they will have to be given a different drug. So this is rare, but not all that rare. A lot of people have been genotyped for their alleles here, and this differs by ethnicity. But overall, quite a large percentage of people are heterozygous. For example, the star 1, star 3A heterozygote is approximately 6.5% of Caucasians, only 1.73% of African Americans, relatively rare in Asians, and reasonably frequent in Hispanics. If you look at the 1C form, only 1% of Caucasians, but almost 9% of African Americans totally. So if you add up all the people who are heterozygotes, those are at risk for developing severe complications. We've got nearly 10% of the population, right? That this is not unusual. This is not a very rare thing. This is something that anybody that's going to be prescribe this drug should be aware of. Now, the 3A, 3C, 3B, etc. homozygotes are relatively rare, but they do occur. They're not that rare. 0.06% of the population is not vanishingly small. And so the effect of this phenotypically is, if you look, approximately 8% of people overall are heterozygotes and will have an intermediate level and therefore are at risk for toxicity from this drug. Okay, so this test, genetic test, is offered as a good thing, but there are many other drugs which people do vary in their responses to, and experimentally, or at least in the clinic, these tests are not always offered. Okay, here's a slide that shows some drugs that are used clinically and some of the enzymes that metabolize those. Okay, so we already talked about the thiopurine S-methyltransferase, which is a mercaptopurine, which will have an enhanced drug effect in some cases, and which will lead to toxicity. Okay, there are other thiopurine drugs as well. Okay, but there's other pairs of metabolizer and drug, which the interaction, or the genetic variation rather, in the drug metabolizing enzyme will have an effect on people that are given the drug. Okay, so the N-acetyltransferase, or NAT2, is the example I'd like to talk about next. And one of the drugs that it acts upon is one called Isoniazid. Okay, so this is an antibacterial drug. It's given for tuberculosis. It's active at administration, but it's also cleared from the body in its acetylated form. The enzyme NAT2 acetylates it, and then it's excreted. Importantly, people can be rapid, medium, or slow metabolizers of this drug, and this determines how much active drug is present in the body at any given time. So people that are rapid metabolizers acetylate it quickly, right? We're talking about acetylation here as an inactivation thing. And as a result, it doesn't persist very long. And this can lead to treatment failure. If your metabolism is so rapid that you never get a therapeutic dose in your body, then it's not going to work for you because you never get to the amount that you need to actually help you. Now, medium and slow metabolizers will retain it in the body longer, and that is a good thing, but it can also produce toxicity depending. Okay, so what this represents is simply looking at a variation in people for how rapidly this drug is metabolized in the body. Okay, so what was done is they took a bunch of people, gave them a dose of the drug, and then monitored 6 hours later how much Isoniazid was actually still there in the body. So as you go from left to right on the bottom axis, the people that are close to the zero, people that are basically, they don't have much of the drug in their body because they've already metabolized it. So the drug does not persist long in those people. On the other hand, the people further to the right acetylate it, which is to say inactivate it more slowly, and they maintain levels of the drug in their plasma for a much longer amount of time. And that genetic difference between people is going to determine, in part, whether the drug is going to work for them or be toxic. Okay, remember, acetylation inactivates it, with the result that the drug does not persist, and that people that acetylate very rapidly will not get their therapeutic doses very likely, and that can lead to treatment failure. But on the other hand, if you're too slow of a metabolizer, you're going to have enhanced drug action, or else you can have toxicity. And in this instance, what that means is liver failure. So that's not a good thing. All right. So the downside of being a slow metabolizer, you can have unwanted side effects because a lot of things are toxic. And in this case, there are serious and even life-threatening liver damage that can occur if the drug persists for too long at higher levels. Okay, so people did an actual experiment to really ask that question, whether if you dose the person according to their genotype, if this avoids the effects. The two effects being treatment failure if the metabolism is too quickly, and liver failure if the metabolism is too slow. So people actually did genotype people and dosed them according to their genotype, and it works out the way you would expect it to do, that genotyping people, adjusting the dose, actually avoided those complications. So it's a good thing that people are willing to then genotype people to try to figure out what the best dose is for them. Right, so the slow metabolizers, they reduced the doses and found that there was much less liver damage, and they increased the dosage for fast metabolizers, and there were fewer instances of treatment failure. So this makes so much sense, and it's an approach that is not as widespread as it should be, but those days are coming. Okay, so in terms of enzymes that are required to activate drugs, the cytochrome P450 proteins are very important in that. Those are liver enzymes that they carry out a wide variety of oxidation and reduction reactions. And these substrates for these many, many enzymes, more than 50 of them in the human genome, they're responsible for metabolizing drugs, but also steroid hormones, fatty acids, xenobiotic compounds that are found, whatever gets in your diet that doesn't belong there. Okay, so one example where genetic variation is a problem that is attributed to a particular cytochrome P450 enzyme is a drug called clopidogrel, and what its job is to prevent blood clots, to prevent heart attacks. Now this is a prodrug and it needs to be activated, and the enzyme cytochrome P450 2C19 is the one that metabolizes it. Now, unfortunately, some people have inactive versions of this enzyme, and therefore they cannot activate the drug. So if they're taking this drug to prevent heart attacks, they might think they're protected, but they're not, because the drug is not metabolized to its active form because of this enzyme deficiency. Okay, this actually comes with an FDA warning that it won't work for slow metabolizers, because people that don't convert it to its active form will not experience a protection from blood clots. Right, so the active drug binds to platelets, preventing them from aggregating. So the drug does not work for people that are deficient in this enzyme. Problem being, people don't necessarily know that they are deficient in this enzyme. Another very important cytochrome P450 enzyme, which metabolizes some 25% of drugs, if that estimation I read is correct, is the 2D6 protein. Now, this is very variable in the population. This is astonishingly variation in this particular protein. Some forms of the gene are basically totally inactive, and if they're totally inactive, it's not going to work at all. Some are decreased in activity, metabolism is slower. Then in the middle, we've got normal people, and then we've got altered enzymes, and then we've got higher enzymes, where you actually have multiple copies of the gene itself. So there's a huge amount of variation in the population on what alleles people have, and inferring that what responses they would have to the drugs that are metabolized by this particular enzyme. Okay, so it's got huge phenotypic variation among people, largely due to genetic polymorphism. So people can be classified as a poor metabolizer, which they have very little or no activity of this particular protein, might be intermediate, might be what's considered normal, or you might have, in some cases where people have as many as 13 copies of this gene, they have enormously higher enzyme, and therefore metabolize its substrates at a very fast rate. Okay, one really good example of something metabolized by the cytochrome P450 is metabolism of codeine. Okay, so codeine is a prodrug. It needs to be demethylated, and the active form in the body is actually morphine. So what happens to the codeine when it enters the body? Some of it's degraded, and some of it's converted to morphine by the action of the 2D6 enzyme. So this reaction is what's going to produce the active form. Some is simply degraded, and it's or never activated at least to form the active morphine and some of its degradation products. So relying on the fact that this enzyme is going to do its job and create some amount of morphine, but it's not going to overdo it in order to cause formation of too much morphine, which is clearly toxic and possibly lethal. Right, so morphine is the pain reliever, but actually only about 10% of the codeine ends up being morphine. The rest is converted or degraded, excreted, etc. The problem here is that ultra-rapid metabolisms produce much more morphine than do normal. And, of course, poor metabolizers don't produce morphine at all. So that's going to cause a huge difference in effects of different people. So morphine is toxic in excess. You will have respiratory depression, and you can even have death. Other issues that occur are nausea, shortness of breath, etc. So there's such a thing as ODing on morphine. So because of these huge variations, we have to worry about giving anyone codeine, actually, because the inactive form of the enzyme will not produce morphine, and there will be no pain relief, so the drug will not work for them at all. More importantly, if you have ultra-active protein, you will produce too much morphine to the extent that an overdose is actually possible. So this is something that everybody should be concerned about, not just for codeine, but for many, many other drugs where there's variation in population leads to very, very different drug responses in different people.